I was about to start it off with saying good morning, but probably not going to be morning when you first watch this, but it might be. Anyway, Tuesday morning, because we didn't have time to completely do this lab in class yesterday. That's why I'm making this. It would be very wise to actually sit here and watch and listen to the whole thing and not fast forward, because the first part of this is going to be me going through the PowerPoints I would have done in class to explain the different things. And yes, you're going to get distracted by my cats. It's morning. They love being out here. That's Osiris, by the way. Anyway, so the PowerPoints, I'm going to be explaining the different types of genetics things, the dominant and recessive, the incomplete / blended, the codominant, and regulatory genes. So all of these are going to be covered during the PowerPoint portion of this. Then we do the papers. So I'm going to start off with giving you the PowerPoint lecture that I would have given you in class. And then I'm going to bring the papers up and show you how to do that. I just realized if I had thought to have my iPad ready, I could also put that on here. So when it comes time for that, I might actually pause this and go grab it, but I'm going to try to just use what's available here first. Yeah. So without further ado, let's get to that PowerPoint. And yes, it takes me a moment because stuff was not centered on my computer screen and I had to fix that because it was driving me nuts. All righty, so here we go. Now, when you look at this online, you're going to see there's actually 3 PowerPoints. The pre-lesson one, just not even bothering with. I talked to you some about that yesterday. The lesson 1 and 2, I put together here. I got rid of the back half of lesson 2 because I don't make you do that anymore and it's not on the homework. I just find it interesting. So if you find any of this interesting, then yeah, feel free to do it. Otherwise, so this PowerPoint in this video covers everything you need. So here we go. And yes, I'm using Harry Potter because hello, Harry Potter. And this is when I find out that so many people in my classes now don't care. Now, as I do this, these are images that I just simply got off. I just Googled. And so most of these pictures actually come from the movie. The movie and the book is slightly different, and the only reason I'm telling you this is for one important fact, and it has to do with Harry Potter's eyes. Harry Potter in the book has a different color eye than Harry Potter in the movie. And the reason is because J.K. Rowling, the author, really liked this kid and thought he was perfect for playing Harry Potter. But he had blue eyes and she did not want to make him wear color contacts. That's the only reason there's a difference in color. So when I'm reading this and we're going over it and I'm talking hair color, it has to do with the books from Harry Potter, just so you know. All righty, here we go. Harry Potter, the reason we use this as an example is because you can actually track all of these inherited genetic traits in the world of Harry Potter. And then when we get to the crazier ones, the blended and the codominants, we use some of the more fantastical characters from Harry Potter. So it's a very easy one to use. I tried to just click ahead and forgot I had touched something else. So here we go. All the Weasleys have red hair, freckles, and more children than they can afford. The red hair and the freckles, totally genetic traits. The more children than they can afford, that's not a genetic trait. So for them, we are going to be looking at the red hair and freckles. He was almost twice as tall as a normal man and at least 5 times as wide. That's Hagrid. He's an example of incomplete or blended traits. And I keep saying incomplete or blended because you can use either name. Incomplete is the one that will be on pretty much everything you see, but it's the same thing as blended, and we covered it last week in that lecture. Harry had a thin face, knobby knees, black hair, and bright green eyes. For him, we're going to be looking at the hair and the eyes. Mostly the eyes is what we're looking at for Harry. A pale boy with a pointed face and white blonde hair. Draco greatly resembled his father. His mother was blonde too. Clearly, we're going for the blonde hair for Draco. Now, here it says, what are some phenotypes described in the 4 excerpts from the Harry Potter books? Well, kind of already told you. We've got the hair color, we've got the freckles, we've got the eye color, and we also have the size, Hagrid. These are all things you can 100% see. So here we go. Here's the 4: freckles, hair color, eye color, height. Now, the genetic traits. These can be described in 2 ways. I've told you over and over again about genotype and phenotype. These are the 2 ways. The phenotype is what you observe. So this is a result of how your genes showcase, basically, how they are expressed. So the phenotype is going to be the hair color. Here it says a talent, sickle cell disease. All of these are phenotypes. The genotype are the actual genes. These are those 2 letters that represent the allele. Yes, if you're going farther in genetics, yes, there's more than that. For us, we're literally just worrying about the 2 letters. So the 2 letters are the genotype. The phenotype is what you observe. Example, freckles. You look at somebody like the Weasleys, they have freckles. You look at Draco Malfoy, he does not have freckles. So has freckles, you can see this. Does not have freckles, you can see this. So the freckles are the phenotypes. The genotype are going to be F. We're using F because F for freckle makes it easy. So a capital F is the dominant letter. That means they have freckles because having freckles is a dominant trait. Then we will have a little f to represent not having freckles. So now, using the big F and the little f, we are going to look at the possible genotypes, so the letters of a pair for freckles. So here we go. Genotype, letters. We have big F, which is homozygous dominant. We have big F and little f, which is heterozygous, because remember, hetero, different, homo, same. And then we have little f, which is homozygous recessive. So now look at the phenotypes. For the homozygous dominant, you see freckles. For the heterozygous, you see freckles. Why? Because you have that one dominant gene in there. Since it is dominant, it will show. And then if you have homozygous recessive, no freckles. This, if you had looked at last week's lecture video, this is an example of the Mendelian ratio. This is for the phenotype, for the genotype, it will be 3:1. We're going to see this in the Punnett Square, which we're coming to in a second. Now, red hair. We're using the letter R for red, so we remember it's red hair. Red hair is recessive. The brown hair would be dominant, but since we are looking for red hair, we are doing the recessive for red. So we have red hair is a little r. Again, recessive, small letter. For brown hair, we need a capital R because that would be dominant, the dominant brown color. Yes, you could use any letters you want. This is just what we're using here. And you get to hear the train. I think you can hear it. I can totally hear it. So using the little r for red and the big R for brown, we are going to look at the various genotypes and phenotypes. Similar to the freckles, we have homozygous dominant. This would be brown hair. We have heterozygous. This would be brown hair. Then we have homozygous recessive, and this is red hair. Now, Punnett Square. This is how we figure it all out. Mom has freckles, dad has none. So each parent has a homozygous genotype. That means both capital or both lowercase. Now, we're going to look at their genotypes. Mom has freckles. So we have to think, what does that mean? Here you go. If mom has freckles, it is homozygous dominant because we already said they're going to be homozygous. There are other options. We'll get to that in a second. But dad not having freckles, he has to be homozygous recessive because not having freckles is the recessive gene. Now we draw this box with lines. That is our Punnett square. And then we put mom across, dad down, or dad across, mom down. Entirely up to you how you do it. But this is what we do. We place them here. And the reason is if you remember in meiosis that mom gives one, dad gives one. And so these are the different ways they can break down. If mom has only 2 capital F's, she can only give a capital F. If dad only has lowercase f's, then he can only give a lowercase f. So you fill it out this way. What you did is you took, let me get the little laser pointer. I would love to know if the laser pointer is actually helpful. I just like using it. You took this F and you brought it down. And then you took this F and you brought it down. Then you take this one from dad and bring it across. And then this F and bring it across. If anybody has ever learned how to use the box method when you are doing foiling in math, you know, the first, outer, inner, last. If you've ever used the box method to do your polynomials, it's the same thing. And if you haven't and you're curious, ask me in class, I'll show you. It makes it so much easier to multiply polynomials. Yes, I'm sorry, I just threw math into your science class. All right, now here's a different one. What happens if both mom and dad have freckles and their genotypes are both heterozygous, which means big F and little f? This is what happens. So if both mom and dad are heterozygous, they have only a 25% chance of having a baby with no freckles, because mom gives off a big F, dad gives off a big F, and then mom gives a little f and dad gives a little f. So there's only one possibility for having a kid with that is homozygous recessive. So this is the Mendelian ratio I was mentioning a second ago of 3 to 1. So when you look at it for the phenotype, freckles, 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 that's 3. No freckles, that's 1. So the Mendelian ratio, 3 to 1. Now, we've just talked about freckles and red hair. Mentioned about the Weasley family, them all having freckles and red hair. So we're going to take a look at that. Now, both parents have to be homozygous recessive. Have to, because mom and dad both have red hair. And if both of them have red hair, they have to be homozygous recessive. So that makes the hair part super easy. The freckles, however, take a little bit more. Since freckles are a dominant trait, we know at least one of the parents has to be homozygous dominant, because if one of the parents was recessive and the other parent, or if both parents were heterozygous, that would be a 3 to 1 ratio. They would have a 25% chance of one of their kids not having freckles. And considering the Weasleys had that many kids and none of the kids came out without freckles, that seems highly unlikely. But if at least mom or dad was homozygous dominant for freckles, that means that parent always gave a capital F, which means the dominant gene was in every single child. So there you go. Now, the Potters. We have Harry, who has dark brown hair. And yes, we just say dark brown hair, even though the quote was black. Harry has dark brown hair like his father's, but his mom had red hair. Now we're going to use the genotypes for red hair that we looked at. We know that mom had to have homozygous recessive because she had red hair. Now, spoiler alert for anyone who is planning to read them and hasn't, or watch the movies and hasn't, we find out that Harry marries Ginny. Ginny, we know, has red hair. She's a Weasley. So we already know what her genotype is. So now we're going to use the Punnett squares to figure out the ratio. So here we go. We know that Harry's mom had to be little r, had to be, because that's the only option because she had red hair. Notice we have two Punnett squares here because we do not know for sure which one dad was because he had the dark hair. He could have been heterozygous or homozygous dominant. Either way, no matter what, we know Harry has dark hair, so he has to be heterozygous for the red hair. So he carries the red hair gene and can pass it. So here we go. They get married and they have babies, which, by the way, the end of the book, the end of the movies, when you see their kids, they actually follow the Mendelian ratio. I thought that was kind of cool. So here we go. We have Harry's genotype, which we just figured out, is heterozygous. Ginny's phenotype is homozygous recessive. So when they have kids, 50-50. Half their kids, well, it's not half their kids, it's just a flip of a coin. Each time they had a baby, it was a 50-50 shot. So 50% chance the baby would have brown hair, 50% chance the kid would end up with red hair. So there you go. This is always the fun part at the end of this. We normally do this in class. I leave it up to you. These are some Mendelian trait examples for people. Tongue rolling, where you go, that is tongue rolling. It is dominant trait if you can roll your tongue. So that means I have the trait, the gene for tongue rolling. Every time I teach this, at least one student in the class cannot roll their tongue. And I always found that interesting. So if you cannot roll your tongue, seriously, let me know because I'm dying to know. Then, of course, we've already talked about freckles. We have widow's peak. Widow's peak is where your hairline kind of peaks down. Earlobes, if they're free hanging, that means there's some space here. This is the dominant trait. Attached means it's like that. And so it like attaches straight here. There's no actual loose lobe. That is a recessive trait. Cleft chin, so where it looks like there's a little line here. George Clooney has that. That is a dominant trait. Hitchhiker's thumb. This is the one I get the most questions about. Hitchhiker's thumb, this. If your thumb goes shoop, that's the hitchhiker's thumb. If when you pull it back as far as you can, it only goes like this, it's straight, that is the recessive trait. Some people have like, it looks like a hook. It's bizarre. And then, of course, we have dimples. So there you go. Now, here's the second PowerPoint, the half that I have, or that I decided to give you. So we have already looked at the Mendelian inheritance of dominant and recessive genes. Now we're looking at the complex traits. These are the ones where we're going to have the incomplete, the codominance, multiple alleles, and regulatory genes. So here we go. Incomplete dominance. We are going to talk about if we had a red flower and a blue flower. If you mix the colors red and blue, you get purple. Last week's video, I used flowers and I said a white flower and a red flower, you get a pink flower. If you just think of this as colors, and it doesn't have to be colors, but if you think of it about colors and you ever did paint or food dye, Play-Doh, stuff like that, imagine mixing these two things together. And that's how you get the middle. Okay, so in Harry Potter, we're going to talk about dragons. That's part of why it makes Harry Potter so easy. So Harry Potter, we have the firepower allele. We have strong fire and no fire. Strong is going to be F. No fire is going to be F apostrophe. Notice they both look exactly the same, but instead of it being capital and lowercase, we have an apostrophe to mark which is the other one. Because remember, this is not total dominance and recessive, so we cannot list them like the dominant and recessive. Here we go. If we have two Fs, no apostrophes, that is strong fire. If we have two that are F apostrophe, that means both of them are no fire, so there's no fire. But if we have one F apostrophe, that's right in the middle. So what is right in the middle of strong fire and no fire? That would be moderate fire. So not strong, but it's not gone. So the blended trait for the fire would be moderate. So not strong, but it is still there. Co-dominance. I normally tell people co-dominance, co-captain, co-ed. It means both are present. Here, red flower and a white flower. You put those two together, you will have a red and white spotted flower because you will see both of them at the same time. So in this example, we're going to use mer people, you know, like mermaid, merman. So mer people, they have tails. We will say you have an allele for a blue tail and you have an allele for a green tail. If you have two B's, it is a blue tail. If you have two G's, it is a green tail. If you have a blue and a G, a blue and a G, a B and a G, then you would have a blue and green tail. Notice I said blue and green. I did not say bluish green. Bluish green would be a blended trait. So for this, blue and green. Multiple alleles. This one I find the most interesting. We're just going to cover the alleles right now and what this means for the genotype and phenotype. The actual blood stuff we cover in Bio 124. So we already know, hopefully most people know, for the phenotypes, for the blood types, we have type A, type B, type AB, and type O. So we have 4 blood types. I, type A. Genotypes, however, we have 6 genotypes. Isn't that weird? We have AA and AO. Genotype AA and genotype AO are both phenotype A blood type. Genotype AB is AB phenotype. That's easy. Genotype OO is phenotype O. Super easy. But then just like the A blood, B blood type has a genotype of BB or BO. This is extremely important because on the complex traits paper, you need to answer questions about this one. So here, if parents have AO and BB blood types, what are the possible genotypes and phenotypes of their children? So we have said that we'll just say mom and dad, it's so much easier. If mom has type A, but her genotype is AO, and dad has type B and his genotype is BB, here are the possible children. 50% chance these parents will have a child with AB blood. 50% chance these kids will be born with phenotype type B blood. So if you are asked for the genotypes and phenotypes, this is what you have to do. You say genotypes and give me the letters, and then phenotypes and what's observed. Now, regulatory genes. Regulatory genes really are not difficult. They just look confusing because it tells you it's regulatory genes. It really works just like the dominant and recessive. So for here, we're going to talk about a Manx cat. A true Manx cat is born with no tail. So what happens here is there is a tail silencing gene. So there is a dominant gene that tells the little developing body, do not make this tail. That's why here they say it's a silence another gene. So basically it just says, no, don't make it. And so here we have the capital S means that it's going to silence the tail gene, which means there is no tail. We have the little s, which means it did not silence the tail gene. And so this cat will be born with a tail. So no matter what its mom is, it's not counted as a Manx because one of the proper definitions of a Manx cat is born without a tail. So the question here is, can two Manx cats without tails, that's kind of redundant, but it's to make sure you remember, have a kitten with a tail? You would have to use the Punnett Square to answer this. If both parents are heterozygous, then yes, they can, because remember the Mendelian ratio of, and I go back for my laser pointer, Mendelian ratio of 3. So these 3 do not have a tail to 1. This one does. So if both parents, there we go. If both parents are heterozygous, it is possible to have a baby that has a tail. Now, Harry Potter, where do these complex traits come from in Harry Potter? Well, Hagrid. He's huge. He's super tall and wide. And then we have Harry's eye color. What would the genotype be for Harry's eye color if he had had any siblings, and what would their eyes be? First, we're going to look at Hagrid. Hagrid was a wizard, or Hagrid's father, sorry, was a wizard. He's actually a weak wizard. We talk about that later, but not in this one because that's the more complex that I don't make you do. So Hagrid's father was a wizard. His mother was a giantess. If the normal height for giants and wizards are as follows, giants can be about 20 feet and a wizard can be somewhere between 5 and 6 feet. Hagrid is 12 feet. They say so in the book and the movie. So what you do is look at this. You take the average of the two. So if you said the wizard is 5 foot and the giant is 20 feet, that is 25 feet. You divide it by 2 because the average is you add the two numbers together and then you divide by how many you had. So here it's going to be 25 divided by 2 gives you 12.5 feet. That perfectly shows Hagrid's height. Yes, they said 12 feet. Giants, it's about 20 feet. Maybe the mom was 19 feet. Maybe she was small for a giant. We don't know. Doesn't matter. It fits. It's right in between the two with that little variation. Then we have Harry's eyes. This gets a little more complex. I just like to show it. So we have Lily had green eyes and the dad has hazel eyes, which is brown and green. So here we go. We had the dad had blue and green eyes. Brown and green eyes. And then the mom has green eyes. So there was a 50% chance that Harry would have hazel eyes, 50% chance that Harry would have green eyes. Now, that's all for this part. It's now time to do the actual lab. So at this point, and yes, make sure I check before you go for credit. Clearly, we're not doing that because you're watching this at home, but you really need to do these papers before Monday because the test is on Monday. So throughout this week, and yes, I'm about to go over the papers. Throughout this week, work on this. When you finish the page, take a picture of it and send it to me and ask me to look over it so I can verify if you're right. I would just do the whole thing for you, but when I cover it, and I don't mean for you, but I mean I would do an example and then let you look at it for correct answers. But since it's randomized, it makes it hard to do that because I don't want to confuse you. And so you have to actually do the lab, but send it to me as fast as you can so I can give you feedback and I can tell you if you're on the right track. You don't even have to finish the whole sheet. If you do half of it and say, I'm getting confused, or I want to make sure I'm right, or I want to make sure I understand it, take a picture, send it to me. I'll look at it. I'll let you know if it's right or not. That way we can do the back and forth feedback. I will do that with you all the way through Friday. Okay. By the time it gets to this weekend, hopefully you will have completely finished this lab, so you are practicing it. And I will be also posting a practice for the test, but you need to do the lab first. Very important. Okay, so that is the end of that part. Now let's look at the actual papers. Okay, which one? This one. We start with the monster genetics lab. For those of you who were in class, I tried talking about this, but it was like the last couple of minutes because we ran late. What you're going to do is flip a coin to have the random assortment, because you're not going to learn anything if you're just doing what everybody else does. So if you actually read the words on this paper, the stuff up here, these are the directions. So you will flip a coin twice to determine the genotype. Remember, the genotype are letters. So I am going to come down here. What I myself do is I put my stuff here. I do heads and then tails. I do this so I don't forget. You do it however you want. I then flip a coin. We're going to pretend I just flipped a coin. Okay. I'm going to say I flipped heads. Since I flipped heads, I then put a big E in the genotype. That's only one gene. So now flip the coin again. And hey, my pretend coin is tails. Tails says this. So I put a little e. This gives me my genotype. And actually, I'm going to put this in the center because I think that just looks better. Actually, I want it more than that. I don't see another option. So I guess that will just have to do. Anyway, I wanted it to be in the middle. So I can totally get distracted way too easily. I know. But here we go. Here's my genotype. Ooh, I know what I do. There. Nope. I can't make it work. Why can I not make it go where I want it to go? That'll just have to do. So that is my genotype. My genotype are the two letters. My phenotype, well, we have a capital and a lowercase. That means it is dominant and recessive. So here, since there is a big E, this phenotype will be 2 small i's. That's what I put there. Now here, we are going to do the eye color. It tells you it is incomplete. So let's say, again, flip the coin. Oops, I have tails. This will be R'. What in the world just happened here? That is weird, isn't it? There we go. Okay, that is R'. Now, flip it again. For the sake of remembering what these things are, let's say it was heads. I, myself, am going to go to this side and do that. So my genotype is R and R'. So if I have R and R' for my genotype, the phenotype would be if I took red and white and mixed it together. If I mix them together, that would be pink. Yeah. If I had done 2 tails, then I would have said white. Okay. So alleles, this is the phenotype, and the genotype is in parentheses. Last example on this side. I'm going to flip that coin, and of course, because I keep doing heterozygous to show you examples, we're going to say heads. So that gives me a G. I flip my imaginary coin and now I have tails. So that would be B. And so this would be green and blue. Remember, and not bluish green, not greenish blue, not green blue, not blue green, but green and blue because of that co-dominant. Please use an actual coin because the likelihood of me doing 3 heterozygous in a row, not quite typical. So this is making mommy monster. Notice it says right here, it says female monster. So this is mommy. When you flip to the other side of the page, look at this. This is daddy monster. Why is this important? Well, because of this. Notice some of this is filled in. You do not change anything that's already filled in. This is letting you know that the genotype for daddy are 2 little e's. Now you have to say, what is the phenotype? Well, let's look back at these traits. If it's 2 little e's, it would be 1 large I. So his phenotype is 1 large I. Eye color, incomplete, white. His phenotype is white. So you look back here. In order to be white, has to be this, which would be R apostrophe R apostrophe. And then you see, you basically do this. Why this is important. Once you have this whole thing filled out, and by the way, when you get to this one, feet incomplete, that is the #1 thing that people get wrong on this homework, on this lab. When you do this, make darn sure you are right, or ask me to make sure you are right, because I'm not kidding. This is the one that people always mess up. Anyway, when you get down here, it says create Punnett Square and attach your work to predict the traits. I have to see your Punnett Square for every one of these. Now, before I show you an example of making the square, I want you to pay attention to the fact that these are not all of the stuff in the chart. So don't just go and think you're done. Pay attention. Here, eyes, what percentage of offspring will have only 1 eye? That's the one we're going to do right now. And this is where I said I kind of wish I had my iPad with me, but we're going to try to do it here. Let's see. Okay. 1 eye, that's little e. And mom has big E, little e. So see, we are taking mom's genotype and we are taking dad's genotype. Whoops. We are taking these 2 genotypes. So now let's see. I'm seeing if I can do more than one thing at a time. No, I can still only do 1 screen at a time. Okay, so I'm going to stop this, then I'll come back to it because I want to do the whiteboard. All right, this is going to be horrible, and I do apologize ahead of time. Let's see. Let's see, draw. Here we go. This is not too bad with that. It'll just be, oh, hey, wait, I might actually be able to do this pretty decently. Okay, so we build our Punnett Square. There we go. And now we have mom. Let's see, I want it to be, yeah, we'll put it there. We have that. And then we have this, because that was mom. Remember, she had 1 big E, 1 little e. And dad was a little e and a little e. Hello, Osiris. My kitty's here. I do hate that it's not exactly where I wanted it. Oh, well, we'll just have to deal, right? So now we bring moms down big E, and then dad's over little e. Then we can bring mom's down, big E, dad's little e. And then we have mom's little e, dad's little e. And then we have mom's little e and dad's little e. So this is our Punnett square. If you look at this, we have a 50% chance of two small eyes and a 50% chance of one large eye. Because see, half is this, half is that. So you would come back. I really wish I could do both at the same time. There we go. You come back here and you have the square that you just drew to prove to me you figured it out. And then what percentage of offspring will have only one eye? That would be 50%. You do this for all of those. You figure out the Punnett square. Okay. Now, last part of this is the other homework page, the complex traits page. So that would be this one. Incomplete dominance, incomplete blended, fire allele. We covered this in the PowerPoint where the F is strong fire or lots of fire and the F apostrophe is no fire. There are more than one question here. There's two questions. I was about to say there are multiple questions and then change what I was saying. You have this one, which is saying, if a dragon has very strong fire, is crossed with a dragon with moderate fire, what would their offspring be like? So here you draw a Punnett square because it says justify your answer with Punnett squares. You draw a Punnett square of a dragon with strong fire and one with moderate fire, and you look at what babies they would have. Then it asks, under what conditions can a baby dragon be born that never has fire? This means you have to use what you just learned about the Mendelian ratios. What blending would give you a baby with two F apostrophes? So give it a try. If you're not sure, try it and then send me what you did and I'll tell you if you are right or not. Codominance, mer people. And let me just go ahead and do this. These are the two most important questions on this complex traits paper. 100% the most important questions on this complex traits paper. So make darn sure you've mastered these two. This one, very similar to what we had in the previous homework, and we went over in the PowerPoint just moments ago, where we have mer people. Cross between two mer people that both have, and that should not be bluish green, that should just say blue and green tails. I don't know why. And I'm fixing it now, even though your paper is going to have something different. There you go, blue and green tails. Give the offspring phenotypes with percentages. This means you have to give me the phenotypes. So like say, and this is not going to be accurate, I'm not drawing it. And you will notice quickly when I give you these phenotypes, it doesn't add up to 100%. I'm going to say 25% blue tail, 25% green tail, 25% blue and green tail. So you have to say the color and the percent. Since you have 4 squares in your Punnett square, it will always be either 25%, well, 0%, 25, 50, 75, or 100. Those are your only percentages possible. 0% means it's not in any of the squares. 100% means it fills all the squares. And then the others would just be 25, 50, well, actually 25, 50, or that, 75, and then 100, of course, is all. Multiple alleles, this is the blood typing. Gave you an example in the PowerPoint moments ago. Here again, you're trying to find out if it's possible to have something happen, and you have to draw a Punnett square to justify. And then regulatory genes, this is just like the Manx. It's not giving you any letters because you can come up with your own letters to use. but you have to actually use the Manx regulatory, the silencing gene example that I just showed you work in order to answer this. So that's the lab. That's the Harry Potter genetics lab. That's how to work them all through. 100% best thing you can do is take your time, be very careful on Mommy Monster, make sure you got it right. Before you switch to Daddy Monster, if you are not 100% positive that you've done it right, halfway through or when you finish Mommy Monster, take a picture, send it to me. When you send it to me, whether it's through Moodle or through regular email, make sure to also text me. I gave you that number at the beginning of the course. Yeah, I'm not going to look it up again. I gave it to you at the beginning of the course. If you have the number, go ahead and text me, because then you can say, hey, I just sent it, and then I can immediately jump over and check. If you don't text me, I will try to keep up. I am going to make sure to check my email and Moodle a couple of times each day this week, just in case you're waiting. If I am not busy when you message me, I will immediately go to it and give you the feedback, because I want you to feel confident in what you're doing. This is a long lab. It's very tedious. It is worth double. So these are a lot of points right here. So you want to get it right. And you want to own this material, because the better you do and the better you understand what you're doing on this homework, the better you will do on exam 3. You have to feel confident in the ratios, in what's different between genotype and phenotype, drawing your Punnett squares, reading your Punnett squares. That is why you have to do so many different things in this homework, is because if you truly do this, you don't just try to Google the answers and you don't ask somebody else to do it. If you truly do it yourself and you ask questions and you get to where you're like, oh my gosh, this is actually kind of easy, because it really is once you know, but almost everything on this planet is really easy once you understand it. Once you've got it, the test on Monday is going to be so much easier. If you just kind of, good enough on this one, it's going to bite you in the butt on Monday. So please, 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 please take the time, really do this. Send me screenshots, not screenshots, but take pictures and send them to me so I can actually give you feedback throughout this week. Once you hit this weekend, you're pushing it too close to the end, and I also cannot guarantee how fast I'm going to reply. So please start ASAP. Okay, I think that's it. I'm hoping I drilled in the importance of this homework. And here's one of the few times this entire term that if you missed class yesterday, you can still do this lab. So please, 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 get it done. And don't forget to practice the DNA lab stuffs one that we did in class, because you'll need to know that one too. But get this one done. All right, that's it. Peace out.